Welcome to the Deep Dive, the show that extracts the most important nuggets of knowledge from our sources, just for you. Today, we're plunging into a topic that affects every single one of us, every single day, whether we realize it or not. I mean, think about it. Your body is a constant construction zone. Every couple of months, all the skin cells on your body are shed and replaced. The lining of your lower intestine, that turns over completely in what, just 3 to 5 days? It's truly remarkable, and it gets even wilder when you look at other organisms. Take newts, for example, famous for their ability to regrow entire limbs. Or the tiny flatworm, planaria, you can actually slice one into numerous pieces, and each piece can form a complete new worm.
Yeah, that's thanks to these specialized, incredibly active stem cells. Amazing stuff.
Right, so this constant renewal, or sometimes a lack thereof, in these incredible regenerative abilities, it all kind of boils down to one fundamental microscopic process: cell division. So for this deep dive, we're embarking on a journey into the intricate world of how cells divide. And our main source today is the fantastic Chapter 14 from Karp Cell and Molecular Biology, 8th edition. Our mission today is to extract the most important insights, the molecular processes, those groundbreaking experimental discoveries, and the real-world relevance from this crucial chapter, making complex biology understandable and hopefully engaging for you.
It's a fantastic mission because cell division truly is, well, the dance of life. It's fundamental to everything from how we grow to how tissues repair themselves. It's happening inside you right now.
So let's start at the very beginning with the cell cycle itself. What is the cellular dance, and what are its main acts?
Okay, so at its core, the cell cycle is the series of defined stages a cell passes through from one division to the next. For eukaryotic cells, that's cells with a nucleus, like ours, there are two main types of cell division we'll talk about. First, we have mitosis. That's all about producing genetically identical daughter cells. Think growth, tissue repair, replacing old cells.
Okay, identical copies. Got it.
And second, there's meiosis. Now, this one's quite different. It produces cells with half the genetic content, and it's absolutely essential for sexual reproduction.
Right, so mitosis for growth and repair, meiosis for making gametes, basically. And what are the major phases of this cycle?
Precisely. The cell cycle is broadly divided into two major phases: the M phase and interphase. M phase is where the actual division happens. That includes mitosis, the nuclear division part, and cytokinesis, where the whole cell physically splits in two.
The main event, so to speak.
Exactly. Now, interphase, that's the period between cell divisions. But it isn't just downtime. You can think of it as the cell's long, intense preparation phase before the big show in M phase. It's further divided into three subphases: G1, S, and G2. The cell grows, duplicates its DNA in S phase, and then makes final preparations in G2.
S for synthesis, I assume. DNA synthesis.
You got it. And actually, a surprising historical insight into this came from studies back in the early 1950s. Researchers used radioactive thymidine, a DNA building block, and autoradiography. And what they found, looking at Karp's figure 14.2, was that DNA replication, the S phase, happens during a specific defined period within interphase. Not continuously, as they might have guessed.
Ah, so it wasn't just happening all through the prep time.
Nope. It showed this incredibly precise timing of events in the cell cycle, even back then.
That's fascinating. So this precise timing, do all cells in our body go through the cycle in the same way, at the same rate?
Not at all. It varies a lot.
Huh.
We can kind of categorize cells into three broad groups based on their division capacity in the body. First, you have highly specialized cells, like mature neurons or muscle cells. Once they're formed, they pretty much lack the ability to divide. They just do their job until they die.
So they exit the cycle permanently.
Right. Then there are cells that normally don't divide but can be induced to if needed. Think of liver cells regenerating after damage, or lymphocytes, white blood cells, multiplying rapidly when you have an infection.
Okay, the on-call dividers.
Sort of, yeah. And finally, there are cells with consistently high mitotic activity. These are often stem cells, like the ones in your bone marrow, constantly churning out new blood cells, or cells in your skin, your gut lining, plant meristems too. And a really key concept here, especially for stem cells, is asymmetric cell division.
Asymmetric, how does that work?
It's clever. A stem cell divides, and one daughter cell remains a stem cell that's self-renewal, keeping the pool going, while the other daughter cell starts down the path to becoming a specialized cell for that tissue.
Ah, so it replaces itself and makes a differentiating cell. Neat.
Exactly. It maintains the balance.
So understanding these mechanisms, from the timing revealed by those early experiments to asymmetric division, it sounds absolutely fundamental. Understanding how the cell cycle is regulated seems key for things like regenerative medicine, but also for, keeping diseases like cancer in check.
Absolutely. Cancer is essentially uncontrolled cell division, so understanding the controls is paramount.
Okay, let's unpack this further then. We know cells divide, but how do they actually control this intricate dance? What are the conductors of this cellular orchestra?
Right. Well, a major breakthrough came from some really cool cell fusion experiments by Rao and Johnson in 1970. Karp shows this in figure 14.3. They fused mammalian cells that were in different stages of the cycle, and the aha moment was when they fused a mitotic cell, one actively dividing with cells in G1S or G2.
And what happened?
The cytoplasm from the mitotic cell forced the nucleus of the interphase cell to do something drastic: premature chromosome compaction. The chromosomes condensed as if they were entering mitosis, even if they weren't ready.
Wow. So something in the mitotic cell was driving it.
Exactly. It was direct proof of powerful diffusible factors in the cytoplasm that actively stimulate mitosis. This led straight to the discovery of maturation promoting factor, or MPF.
MPF. Okay. What is it made of?
MPF, first found in frog eggs, turned out to have two crucial parts. One is a kinase, remember. That's an enzyme that adds phosphate groups to other proteins, acting like a molecular switch. And the other part is a regulatory protein called cyclin.
Ah, cyclin.
Yeah.
Here's where it gets really interesting, right? Because of the name.
Precisely. It's called cyclin because its concentration cyclically rises and falls in a predictable pattern with each cell cycle. This cyclical change is what dictates MPF's activity. Figure 14.4 in Karp illustrates this beautifully.
So the amount of cyclin controls the activity of the kinase partner.
You've got it. And this tied in perfectly with Tim Hunt's 1983 discovery. He found proteins in sea urchin embryos whose levels dramatically increased and then plummeted with each division. These were the cyclins.
So Hunt found the protein that cycles, and that connected back to the MPF activity. It all came together.
Exactly. And eventually, MPF was purified and identified as a cyclin-dependent kinase, or a CDK. The kinase part depends on its cyclin partner to function. And even more remarkably, the CDK subunit was found to be basically the same as a key cell cycle protein already known in yeast called CDC2, showing this incredible evolutionary conservation.
Wow. From yeast to frogs to us. The basic machinery is the same.
It really is. These CDKs are the drivers or orchestrators. They phosphorylate tons of different proteins at just the right times to push the cell through the cycle stages.
But they can't just be on all the time, right? You mentioned controls.
Right. They're not just always on. Their activity is super tightly regulated by a whole system of brakes and accelerators. First, obviously, is cyclin binding. The CDK enzyme is inactive until the right cyclin builds up and binds to it. That binding changes the CDK's shape and turns it on.
Okay, so cyclin level is the first control. What else?
Then there's CDK phosphorylation and dephosphorylation. It gets a bit complex, but think of it like more switches. For a CDK to be fully active, it usually needs an activating phosphate added by an enzyme called KPIC. But there's also an inhibitory phosphate that can be slapped on, often by an enzyme called WEE1. This inhibitory phosphate keeps the CDK turned off, even if cyclin is bound.
So it needs an activating phosphate, but it can be shut down by an inhibitory one.
Correct. And the go signal often comes from another enzyme, a phosphatase called CDC25, which removes that inhibitory phosphate. When CDC25 acts, the CDK is finally active and can drive entry into mitosis.
Okay, so KIC activates, WEE1 inhibits, CDC25 removes the inhibition.
Exactly. And the phenotypes of mutant yeast cells, shown in figure 14.6c, really nailed this. If you mutate WEE1, you lose the inhibitor, so cells divide too early, they become wee or small. If you mutate CDC25, you can't remove the inhibitor, so cells can't divide and they grow really large. It's striking visual proof.
Wow, that makes it clear. Are there other breaks?
Yes, there are also CDK inhibitor proteins, CKIs. Proteins like P21 and P27 can bind directly to the CDK cyclin complex and block its activity. There's a really compelling experiment, figure 14.0BC, with P27 knockout mice. Mice engineered to lack P27 grew significantly larger than normal, with overproliferated organs.
So removing that one break led to too much cell division.
It clearly showed P27's critical role as a brake in controlling cell growth and differentiation.
Okay, breaks and accelerators. What's a major accelerator?
One of the most crucial accelerators, which also ensures the cycle moves forward irreversibly, is controlled proteolysis, targeted protein destruction.
Destroying proteins makes things go faster.
By eliminating key inhibitors or proteins that hold the cell back. The main player here is a big molecular machine called the anaphase promoting complex, or APC. The APC acts like a tagging machine. When it's time for anaphase, The APC, with a helper protein called CDC20, tags another protein called securin for destruction by the cell's garbage disposal, the proteasome.
Okay, so APC tags securin. What does securin do?
Securin normally holds an enzyme called separase inactive. So when securin is destroyed, separase is released. And separase is the enzyme that cuts cohesin. Remember, that's the molecular glue holding the duplicated sister chromatids together.
Ah, so APC destroys securin, which frees separase, which cuts the cohesin glue.
Which triggers the sister chromatids to separate, starting anaphase. It's a direct molecular cascade. Later, the APC, with a different helper called CDH1, also targets the mitotic cyclins for destruction, causing the cell to exit mitosis.
That destruction part sounds really final. How do they show it was irreversible?
Yeah, that irreversibility is key. There was this really elegant reverse mitosis experiment, figure 14.27. Scientists arrested cells in mitosis using drugs that block protein destruction, proteasome inhibitors. Then they chemically inhibited CDK1 activity. The cells actually exited mitosis chromosomes, decondensed, nucleus reformed, even without protein destruction.
So they went backwards.
Temporarily. But here's the kicker. When they removed the CDK1 inhibitor, allowing CDK1 to become active again, the cells actually re-entered mitosis because the mitotic cyclins hadn't been destroyed.
Whoa. So without destroying the cyclins, the cell could actually go back into mitosis.
Exactly. It powerfully demonstrated that proteolysis, the destruction of key proteins like cyclins, is what normally prevents this backward movement and makes the cycle strictly one way. And just briefly on mammalian CDKs, figure 14.8b shows we have several, but studies found that CDK1 is actually the only one absolutely essential for division. There's some redundancy, but CDK1 is king.
Wow, okay. So many intricate controls. It's like a high security system. Which brings us to what happens if something goes wrong? Like DNA damage. How does the cell ensure accuracy?
That's where cell cycle checkpoints are absolutely critical. Think of them as surveillance systems. They monitor the state of the cell. Is the DNA okay? Is replication complete? Are chromosomes aligned? And they can halt the cycle if something's amiss.
Quality control stations.
Exactly. They ensure fidelity and prevent the propagation of errors, which is crucial for preventing things like cancer. People with defects in checkpoint genes, like in ataxia-telangiectasia, have a much higher cancer risk.
So how do they work? What if DNA gets damaged?
There are well-studied pathways, shown in figure 14.9. Let's take two examples. If UV radiation damages DNA, sensors activate a kinase called ATR. ATR then activates another kinase, CHK1. CHK1 then phosphorylates and inactivates CDC25, remember, the enzyme that removes the inhibitory phosphate on CBK1.
Right, the one that lets the cell enter mitosis.
So if CDC25 is inactivated, CDK1 stays inhibited, and the cell arrests in G2, giving it time to repair the UV damage before attempting mitosis.
Clever. And what about other types of damage?
For damage like double-strand breaks caused by ionizing radiation, a different kinase, ATM, gets activated. ATM activates GH2, which then phosphorylates and stabilizes a super important protein called P53. Normally, P53 is very unstable, but when stabilized, it acts as a transcription factor, turning on the gene for a CDK inhibitor protein, P21.
The CKI we mentioned earlier.
Exactly. So P21 levels go up, it binds and inhibits G1 CDKs, and the cell arrests in G1, preventing it from replicating damaged DNA in S phase.
And P53, that name rings a bell in relation to cancer.
Hugely important. It's mutated or inactivated in something like 50% of all human tumors. It's often called the guardian of the genome because of this checkpoint role. Its loss is a major step towards cancer.
Incredible. Okay, so the checkpoints guard the gates. Assuming everything is okay, we get to mitosis itself, the actual choreography of division. How does that unfold?
Right. Mitosis is the process of segregating those duplicated chromosomes equally into two new nuclei. It's typically divided into 5 stages: prophase, prometaphase, metaphase, anaphase, and telophase.
Let's walk through them. Prophase is first. What happens there?
Prophase is mostly about getting the chromosomes ready. They undergo massive compaction, becoming much shorter and thicker, so they're manageable units. You can see this in figure 14.12. This compaction involves a protein complex called condensin, which helps loop and coil the DNA.
And the duplicated chromosomes, the sister chromatids, are still stuck together.
Yes, they're held tightly together by that cohesin complex we talked about, acting like rings around the DNA. Figure 14.18. During prophase, cohesin is actually removed from the arms of the chromosomes, but it stays put at the centromere, the constricted waist region.
Okay, so they're joined only at the centromere by late prophase. And the centromere is where the action happens for movement.
Pretty much. The centromere is where a complex protein structure called the kinetochore, assembles on each sister chromatid, figure 14.1. The kinetochore is vital. It's the attachment site for spindle microtubules, it houses motor proteins, and it's part of that checkpoint system.
Right. And the spindle, how does that form?
In animal cells, it starts with the centrosome cycle, figure 14.1. The centrosome duplicates earlier, and in prophase, the two centrosomes move apart towards opposite sides of the nucleus. As they move, they start nucleating microtubules, forming star-like structures called asters. These migrating centrosomes establish the two poles of the developing mitotic spindle, figure 14.1.
So the centrosomes set up the poles.
Usually, yes. But what's fascinating and highlighted in Karp figure 14.1 is that some cells, like plant cells or even early mouse embryos, can build a perfectly functional bipolar spindle without centrosomes at all.
Really?
They seem to use proteins on the chromosomes themselves to nucleate microtubules, and then motor proteins organize and focus these microtubules into poles. It shows incredible flexibility cells have, as Karp puts it, two fundamentally different mechanisms to achieve the same outcome.
Nature finds a way. Okay, so after prophase comes prometaphase.
Yes. Prometaphase begins abruptly when the nuclear envelope breaks down. Now the spindle microtubules can access the chromosomes. What follows is a dynamic search and capture process. Figure 14.2. Microtubules grow and shrink, probing randomly until they manage to capture a kinetochore.
And once they're captured,
The chromosomes start getting pulled back and forth. It's a tug of war between microtubules pulling from opposite poles. This movement eventually leads to congression, all the chromosomes aligning neatly at the metaphase plate, an imaginary plane equidistant from the two spindle poles. That signals entry into metaphase.
metaphase seems like a pause where everything is lined up perfectly.
It looks static, but it's actually incredibly dynamic. Even in metaphase, there's a constant poleward flux of tubulin subunits through the spindle microtubules, shown in figure 14.25. Subunits are added at the kinetochore end and removed at the pole end, like a tiny conveyor belt.
Huh. Why do that if the chromosomes aren't moving?
It's thought to maintain tension on the kinetochores, which is important for the checkpoint signaling, and it keeps the whole structure primed and ready for the next stage.
Which is anaphase, the big split.
Exactly. Anaphase begins suddenly when that separase enzyme cleaves the cohesin, holding the sister chromatids together at the centromeres. The now separated sister chromatids, now considered individual chromosomes, start moving towards opposite poles. We distinguish anaphase A, which is the chromosome-to-pole movement driven by shortening of the kinetochore microtubules,
Okay.
and anaphase B, where the spindle poles themselves move further apart, driven by motor proteins pushing overlapping polar microtubules and pulling on astral microtubules.
So the chromosomes move in and the poles move out. How do the chromosomes actually move in anaphase A? What pulls them?
It's a really elegant mechanism, actually. Pioneering work by Shinya Inoue back in the 1960s suggested that the force comes from the depolymerization of the microtubule itself right at the kinetochore, figure 14.29. The idea is that as the microtubule shrinks, it pulls the chromosome along. Specific protein complexes at the kinetochore, like the NDC80 complex, figure 14.30, act as couplers, harnessing the energy released by microtubule disassembly to move the chromosome.
So the microtubule falling apart pulls the chromosome. That's counterintuitive, but cool.
It is. And of course, the cell needs to be absolutely sure everything is ready before this happens. That's the job of the spindle assembly checkpoint, SAC.
Another checkpoint. What does this one check?
In SAC figure 14.31 monitors whether all chromosomes are properly attached to the spindle and under tension. If even one kinetochore is unattached or improperly attached, it sends out a wait signal.
A wait signal?
Unattached kinetochores recruit proteins like MAD2, which then diffuse away and inhibit the APC, the anaphase promoting complex.
Ah, so it stops the APC from tagging securin for destruction.
Exactly. This prevents separase activation and cohesin cleavage, delaying anaphase until all chromosomes are correctly attached and aligned. The SAC can also help correct faulty attachments, like when both sister kinetochores attach to microtubules from the same pole, syntelic attachment, figure 14.20b, often using kinases like Aurora B to destabilize incorrect attachments until the right ones form.
Amazing fail-safe. Okay, chromosomes arrive at the poles. What's the grand finale?
That brings us to telophase and cytokinesis. In telophase, the separated chromosomes arrive at the poles, they start to decondense back into their more relaxed state, and new nuclear envelopes reform around the two sets of chromosomes. Mitosis, the nuclear division, is essentially complete.
And we should probably mention the motor proteins again briefly.
Oh, absolutely. As Karp summarizes nicely, different microtubule motor proteins, kinesins, and dyneins are crucial throughout mitosis. They help separate poles in prophase, move chromosomes in prometaphase, drive pole separation in anaphase B, and focus spindle poles. They're the real workhorses.
Okay, so the nuclei are reformed.
Yeah.
Now, the cell itself has to divide cytokinesis. How does that happen in animal cells?
In animal cells, cytokinesis typically starts during anaphase. You see an indentation forming on the surface called the cleavage furrow, figure 14.32. This furrow deepens and constricts the cell in the middle. The mechanism is explained by the contractile ring theory, figure 14.35a. Just beneath the plasma membrane at the equator, a ring of actin filaments and the motor protein myosin II assembles, figure 14.36.
Like a little muscle ring.
Exactly like that. Myosin II pulls on the actin filaments, constricting the ring like a drawstring bag, pinching the cell in two. A small GT paste called Rho-OA is key in organizing and activating this ring.
And then it just pinches off completely.
The final separation involves a structure called the midbody, figure 14.34, and the actual membrane fission event, called abscission, uses specialized machinery, including ESCRT complexes, to cut the connection. Importantly, the position of the mitotic spindle earlier determines where this cleavage furrow forms, ensuring the chromosomes get divided equally.
Makes sense. But plants have that rigid cell wall. They can't pinch like that.
Correct. Plant cytokinesis is quite different. Because of the cell wall, they build a new wall from the inside out. It involves forming a structure called the cell plate, figure 14.38. Vesicles derived from the Golgi apparatus, containing cell wall materials, are transported along microtubules of the phragmoplast remnants of the spindle to the cell's equator. These vesicles fuse together, starting in the middle and growing outwards, eventually forming a complete partition that matures into a new cell wall separating the two daughter cells.
Inside-out wall building, very different. So that covers mitosis beautifully. But you mentioned meiosis earlier, the division for sexual reproduction. Can you give us the key differences?
Sure. The main goal of meiosis is twofold: reduce the chromosome number by half, from diploid to haploid, to make gametes like sperm and eggs, and also to generate genetic diversity. The key difference from mitosis is that meiosis involves two sequential nuclear divisions, meiosis the first and meiosis the second. But crucially, only one round of DNA replication beforehand, as shown in figure 14.39.
Two divisions, one replication. How does that have the chromosome number?
In meiosis the first, the homologous chromosomes, the pair you inherit, one from each parent, pair up and then separate from each other. The sister chromatids stay together. This is the reductional division. Then in meiosis the second, it looks much more like mitosis. The sister chromatids separate. The end result is 4 haploid cells, each genetically distinct.
And this happens at different times in different organisms.
Yeah, the timing varies.
That's right.
Figure 14.40. In animals like us, it's typically gametic meiosis, happening right before fertilization to produce sperm and eggs. Figure 14.41 shows spermatogenesis versus oogenesis. Fungi often do zygotic meiosis right after fertilization. Plants have sporic meiosis at an intermediate stage.
Okay, fascinating. So, bringing this home, what does this all mean for us? What are the human connections, especially when things go wrong in meiosis?
This is where the precision we've been talking about becomes starkly relevant. Errors in meiosis, particularly the failure of chromosomes to separate properly, a process called non-disjunction, illustrated conceptually in the text, e.g. figure 1, are surprisingly common.
Non-disjunction. So chromosomes stick together when they shouldn't.
Exactly. Either homologous chromosomes fail to separate in meiosis the first, or sister chromatids fail to separate in meiosis the second. This leads to gametes with an incorrect number of chromosomes, either too many or too few. This condition is called aneuploidy.
And what are the consequences of aneuploidy in humans?
They're often very severe. Most aneuploidies, like having only one copy of an autosome, monosomy, or having 3 copies, trisomy, of most chromosomes, result in miscarriage, often very early in development. However, there are exceptions. The most well-known is trisomy 21, having 3 copies of chromosome 21, which causes Down syndrome, phenotype shown in figure 2. Individuals have characteristic features and varying degrees of intellectual disability and health issues.
And there's a known link between Down syndrome risk and the mother's age, right?
Yes, a very dramatic correlation. The risk of having a child with Down syndrome increases significantly, especially after age 35. And studies show most of these errors originate during meiosis in the mother's developing egg cell, oocyte.
Why would older eggs be more prone to errors?
The exact reasons aren't fully understood, but hypotheses include the long-time oocytes are arrested in meiosis from before the mother's birth until ovulation decades later, potential weakening of the cohesin holding chromosomes together over time, or issues with checkpoint function.
Interesting. Are there other common aneuploidies?
Yes, aneuploidies involving the sex chromosomes are also relatively common and generally less severe than autosomal ones. Examples include Turner syndrome, females with only one X chromosome, XO, and Klinefelter syndrome, males with an extra X chromosome, XYY.
So errors in meiosis have major health consequences. But meiosis also does something incredibly important correctly, right? Generating diversity.
Absolutely. Beyond just having chromosome numbers, meiosis is crucial for genetic recombination, also known as crossing over. This is where homologous chromosomes physically exchange segments during meiosis thirst.
Swapping bits of DNA between the maternal and paternal chromosome.
Exactly. This shuffles the alleles, different versions of genes, on the chromosomes, creating new combinations that didn't exist in either parent. It's a massive source of genetic variation, which is fundamental for evolution and adaptation. It's why siblings, except identical twins, are genetically unique.
How does that swapping actually happen at the molecular level?
It's an incredibly precise molecular dance, detailed in Figure 14.47. It typically starts with programmed double-strand breaks made in the DNA by an enzyme called SPOA1. Then one of the cut strands invades the DNA of the homologous chromosome, guided by proteins like RAD51. This leads to the formation of cross-shaped intermediates called holiday junctions. These junctions can then move along the DNA and are eventually resolved or cut in specific ways. Depending on how they're resolved, you can get either a full crossover, a reciprocal exchange of large chromosome segments, or a non-crossover, where there might be just a small local exchange of information. The precision is astounding, ensuring no DNA is gained or lost.
Wow. Programmed DNA breaks that get repaired in a way that shuffles genes. That's intricate. What an incredible journey we've taken today, from the constant renewal happening in our own bodies right now to the intricate molecular choreography Karp lays out for cell cycle control and division. This deep dive into mitosis, meiosis, the checkpoints, the cyclins, and CDKs, it really underscores how foundational this knowledge is.
It really does. Understanding these processes is critical for comprehending growth, development, aging, and diseases like cancer and inherited genetic disorders. It even informs how we think about evolution.
It connects us so much.
Absolutely. And if we connect this to the bigger picture, despite the incredible detail and precision we've discussed, scientists are still unraveling subtle nuances. How exactly do all these molecular interactions achieve such fidelity, cycle after cycle? There's always more to learn about how life replicates itself and how these ancient mechanisms, conserved from yeast to humans, hold keys to our health and maybe even future therapies.
A constant process of discovery about a constant process of life. Thank you for joining us for this deep dive into the world of cell division. We hope you feel a bit more connected to the amazing microscopic events keeping you going.